We're going to have exam 2 will be a week from today. I think someone pointed out maybe on an earlier slide, lecture, whatever, I said the exam was going to cover chapters 6 through 11. It's 6 through 10, just as I believe it is in the syllabus. So we're going to finish chapter 10 today. That'll bring us formally to the end of the material for exam 1 or exam 2. Probably get started on chapter 11, which is really an extension of the material we're talking about in 10. Then hopefully we'll get close to finishing up chapter 11 on Monday. I will leave 15-20 minutes for any questions related to the exam, and we'll be right on schedule at that point. Any general questions? Okay. So before we get started, or did I not? Let me see. Hang on just a second. I think I see. I think it's. Oh. Okay. Okay, what I was looking for, I thought I had them. I guess I forgot the 2 starting questions. So if you answer all the rest of the questions that we have for today, then I'll just give you your 20 points, okay? All right, so we're in chapter 10. We're getting into the molecular biology now, and we're going to be, so we've moved away from the more traditional genetics, and we're getting into the molecular aspects of it. So as we start going through these different topics involving DNA metabolism, that is looking at replication, probably something you haven't seen too much, repair, recombination, although we're probably not going to have time to look at that at the molecular level, transcription, translation, and such. As I said last time, you're probably going to see these same topics in other courses, but you're getting it from a different perspective. The biochemist should be more of an enzymology, biochemistry side. My emphasis is going to be more on structure and function. That is, structure dictates function. So as I go through this, for instance, I'm not going to ask you to memorize the structure of each of the nucleotides. I leave that to the biochemists. That's what they get paid to do. But rather, I'm going to point out, how is this molecule different from this molecule in the context of having to be able to talk about that later on when we're talking about replication and some of the other molecular events that are going on. So we started this chapter with really 2 of 3 landmark or classic experiments that unequivocally showed that DNA was the molecule in which genetic information was encoded. So as I said previously, up until the time of these experiments, which was in the 1940s, 1950s, that sort of thing, the preponderance of the scientific community thought that proteins were going to be the genetic material. Although certain things like how to replicate the molecule without causing any errors, trying to come up with those kind of models with proteins doesn't work very well, but people were trying. So to really convince you that this, what was boring DNA molecule, a little bit later we'll see some pictures and talk a little bit about Watson and Crick's work on putting together how they took the data from all these different labs and came together with this model. At the time, Crick was a graduate student and almost got thrown out of his lab because he was wasting his time on this boring molecule and not working on his thesis project. Okay. So we talked about Griffith's experiment, where you take the heat-killed virulent bacteria, mix it with the avirulent or harmless bacteria that are living, inject it into a mouse, and get the unexpected result that the mouse dies. And you isolate bacteria that are now genetically transformed and now have the ability to be virulent bacteria. So this was this transformation, that something from the dead cells transformed the living cells. It took 10 years and Avery, McLeod, and McCarty's work to show that that was in fact DNA. So that was what we did last time, where you use the different enzymes, the proteases to degrade protein, DNase for DNA, and RNase for RNA, and showed that when you treated with the DNase, you lost the transforming activity. Then we got started on this third experiment, Hershey and Chase's classic experiment, where they're taking a different approach to the question. And this is the one way I like doing science is, if your idea is correct, you can come at the problem in several different directions, and it should all converge on the same answer. So Hershey and Chase did their experiment with a bacteriophage. So as I mentioned, I'll just go through this quickly of what we did last time, and then take you into the science and the conclusions. So they're working with this bacteriophage T2. Its biology is very similar to bacteriophage lambda, which was the example of the temperate phage that we talked about. So what they did know at the time, back in 1952 when they published the paper, they knew that if you had isolated phage, all you had was protein and DNA, so there was no RNA present. All right. They knew that the phage attached to the surface of the cell, and since the phage were assembled inside the cell and then lysed the cell, whatever the genetic material was, it had to be transferred from the phage into the cell. So the question was, is it protein that's getting transferred into the cell, or is it in fact DNA? So they designed this experiment, and as I said before, at that time and up until maybe 10, 15 years ago, radioisotopes, radioactivity was used to label these molecules so that we could follow them through the experiment. So they infected E. coli with the bacteriophage and added P32 orthophosphate or inorganic phosphate, and that way this radioactive label would get incorporated into what molecule in the phage? Does it go into protein? No, it would label just the DNA. So you have this preparation labeled with P32 phosphate, which means only the DNA is radioactively labeled. In this sample, they did the same experiment, but they put in S35 or radioactively labeled sulfur, which gets incorporated into what? Into the protein. So you got one set of bacteriophage where the protein is radioactively labeled, and the other, the DNA is labeled. Now all you have to do is figure out which one of those molecules is going to get into the cell. So they purify the bacteriophage, they infect the cells, there's no radioactivity, so the only source of radioactivity is from these radio-labeled bacteriophage. So this is in a cartoon form what these phages look like. So it's a rather complicated protein structure. So this is this icosahedral head. The DNA is packaged in here, and then all of this apparatus is really intended to do one thing. Now we know that the DNA is going to get transferred through this shaft portion of the phage into the cell. At the time, they did not know this, so that was what this experiment is really all about. So you start out with the phage attaching, and so here is the DNA molecule. These little fragments are the bacterial chromosome, because one of the first enzymes being made from the bacteriophage gene is an enzyme to degrade the host genome. So this cell is going to die anyway. Then it begins to package up the phage, and then you lyse the cells, and out they come. So this is the life cycle it's going to go through. The question is, what is this molecule? Is this DNA or is this protein? And that's what this experiment was designed to test. Okay. So they made the cells labeled with the S35, so that would label the proteins. This would label the DNA. All right. Then you use those to infect cells. Here's that high-tech instrument called a blender. The purpose of this is they knew the phage were attached to the cell surface, so they wanted to put enough shear forces in there to literally throw whatever didn't get transferred into the cell was going to get thrown off the surface of the cell. So they shear off these protein coats, and then collect the cells. Now, the bacteriophage are so small that in a typical centrifuge, they're not going to pellet, in the jargon. They're going to remain in the supernatant. So when you centrifuge at fairly low speeds, the bacterial cells will be down here in the cell pellet. The bacteriophage, whatever's left of them, whatever got thrown off the surface of the cell, will be left in the supernatant. And what they found was the S35 radioactivity was in the supernatant, whereas the P32 was in the cell pellet. So what does that tell you? So it was the DNA that got transferred into the cells, and in fact, they even found that some of the progeny phage that were released after the cell was lysed still had some of the radioactive DNA. So the same DNA that had been injected into the cell was getting reused and packaged into another phage. Okay, but this gave us now another line of evidence that says DNA is the genetic material. So that's what okay, and not protein. So that pretty much at that point put the whole question to rest, and the idea that proteins would encode the genetic information finally died the death that it deserved. Okay. All right. So any questions about that? All right. Let's see if. So. All right. Let's try this question then. Okay. Keep in mind this is a hypothetical question. Not. I'll go to 1 minute and 15 seconds on this one. Okay. So what's the answer? All right. Okay. So again, this is a hypothetical question. The other is just to use this as an example of looking at the difference between real data and what's in the textbook. The way this is presented, it looks like it's a nice clean experiment, and only P32 was found in the cell pellet, and only S35 was in the supernatant. If you go back and look at their original paper, you'll find that it's not quite that clean, but they did come to the correct conclusions. Okay. So those were just some of the classic experiments to prove that DNA is a genetic material. That's an interesting molecule and worth pursuing in terms of studying it. What we want to do for the rest of this chapter and what's in chapter 11 is to start looking at the structure of the DNA. I'll point out what parts of it are important for the things that we'll be discussing. And we're going to look at it at 3 levels. The primary structure, which is just really going to be the sequence of nucleotides. The secondary structure, which is this double-stranded helical structure that you're somewhat familiar with, that'll be the end of chapter 10. Beginning chapter 11, which begins the material for exam 3, is we'll look at higher level tertiary structure of the DNA molecule. But we might start on that today. Okay. So Watson and Crick are probably two Nobel laureates that even the average non-scientist recognizes their names. One of the things I'd like to point out here is, especially if you've been taking organic chemistry, I assume you still do these 20-page lab reports, tell me the history of the spectrophotometer and all this sort of stuff. Okay. A couple of things. One, as a professional scientist, whether you're writing a manuscript or a grant, whatever we're doing now, there are very strict page limitations. So you have to be very concise and very precise in your writing. And the second is, it's the content and not the length of the paper that's important. So this Nobel Prize winning paper that was published in Nature back in 1953, in Nature, one of the most prestigious scientific journals for publishing, was in fact one page. So just a one-page paper ended up leading to the Nobel Prize. The other is they've been criticized because they didn't really do any of their own experiments. Although I'm in the camp that believes that this is still good science. They took the ideas from several other people, put them all together, and made it into this important model for us understanding the structure of DNA. So here are the players. A couple of kind of interesting things. Rosalind Franklin did the X-ray crystallography that was really critical to understanding the double helical structure of the DNA. There's been a lot of papers or a lot of criticism as to why she didn't share in the prize. Maurice Wilkins, it was really his laboratory that Franklin did all of her work. He did share the prize with Crick and Watson. As far as three of them have passed away. As far as I could tell, Watson is still alive, but he's about 87 years old now, but he's still active. The main thing is, as I said, Wilkins shared in the prize, but nobody ever seems to include his name. But if you go formally on the prize, his name is included. And Rosalind Franklin wasn't included because there's a rule that you cannot win the award posthumously. Unfortunately, she passed away from cancer at a very young age. Okay. Also, there's a prize that mathematicians are not eligible for the Nobel Prize. Does anybody know why? No, I'm sorry, what? No, it's more personal.
They have the field level instead.
Well, because they have a different one. Yeah, at least the story. I don't think it's an urban legend. I think it's true. Apparently, when Alfred Nobel was setting up the prize, his wife ran off with some grad student in mathematics, something like that. So he stipulated that mathematicians would not be eligible for the prize. So they have their equivalent prizes, but it was personal. Okay. Also, hopefully you guys haven't been undergrad since 2005. But FAU started a Nobel laureate lecture series in 2005, and Jim Watson was actually the first speaker. Was the first speaker. So it was one of the few speakers that it was open to the public, and we ended up having to basically webcast it into several different rooms because the tickets were given out very quickly. So we had 500, 600, 700 people showed up for that lecture. He's an interesting character. I was reading, he was at Harvard, and apparently he was, what I was reading said he wanted, this was back in 1953, he wanted a $1,200 raise for winning the Nobel Prize. And the dean went, nah, not worth it. Okay. So they put this story together, and so what we'll do is I'm going to show you some of the pieces and show you at the end how they took data from several different laboratories and came up with this double helical structure. So as I said, we're going to talk about the primary structure, which is just the nucleotide sequence. The secondary is the helical structure, and then chapter 11 gets into really chromatin organization and higher levels of organization. All right. So most of this you should have some level of familiarity, trying to bring everybody to the same point, and I will be emphasizing those parts of the molecule that are important for what we're going to be talking about. So as we go through the course, we're going to talk about DNA, RNA, and protein. At its most fundamental level, these are polymers. They are made up of subunits, repeating subunits. Each one has its own particular characteristic covalent bond that simply joins one subunit to the next. So DNA is just simply a polymer. And it's composed of these repeating subunits called the nucleotides. In terms of the size of the molecules, DNA is by orders of magnitude larger than any other molecule within the cell. So if you look at a fairly large cellular protein, it has about the same mass as a very short little tiny DNA fragment. Many of these molecules, especially in chromosomes, can be tens of millions of nucleotides in length. So it's a very large, very long molecule, but also extremely fragile. So when isolating it, at least if you're working with the very, very large molecules and trying to keep them intact, you have to take very special precautions not to break the molecule. Okay. So there's going to be three basic components to those nucleotides. You're going to have the nitrogen-containing base, which is either the purine or the pyrimidine, a pentose sugar being a 5-carbon sugar, and then there's going to be phosphates attached to it, either 1, 2, or 3 phosphates. For the sugar, it's a pentose sugar because it's 5 carbons. You've probably heard the terminology 1', 2', 3'. That prime designation is there to tell you that the number refers to the carbon atoms in the sugar ring. The bases themselves, the purines and the pyrimidines, also have another numbering system, but they don't have the prime. So that's why the prime is important. That tells you you're talking about the sugar. You take carbons 1' through 4', join them together with an oxygen, and you get the pentose sugar ring. Okay? And then the 5' carbons joined at the 4. So the main things I want to point out, so here's the ribose, deoxyribose. Which one's in DNA? The deoxyribose. Okay. So biochemically, the only difference between this ribose for RNA and this deoxyribose for DNA is the presence of this hydroxyl on the 2' carbon. Now it's important for a couple of reasons. In some ways, this sterically hinders the RNA from taking on some of the double-stranded structure that DNA has, but it also makes the molecule much more biochemically labile. So DNA is a remarkably stable molecule. This is why we've been able to clone and sequence DNA from, Egyptian mummies and other organisms that have been dead for tens of thousands of years, millions of years, whatever the number of zeros are at the end of it. RNA is not that stable, but RNA is a transient molecule. The cells they constantly turn over and make new RNA molecules. But for right now, the difference between the ribose for RNA and the deoxyribose is the presence or absence of this hydroxyl group on the 2' carbon. The 1' is important because that's in all of these, both DNA and RNA, this is where the purine and the pyrimidine bond is formed. The bond that holds them, the glycosidic bond that holds the base. So the purine and the pyrimidine is attached to carbon 1'. So we got 2'. 3' is critically important. In DNA and RNA synthesis, the new nucleotide is going to get added to this hydroxyl group. If this hydroxyl group is missing, it's called a chain terminator, and synthesis will stop. Some antiviral drugs and anti-cancer drugs, which are really designed to try to stop rapidly dividing cells from proliferating, will use analogs like this, except they lack the hydroxyl group. Those analogs get incorporated into the DNA or the RNA, and then synthesis stops. So you must have this 3' hydroxyl for DNA and RNA synthesis to occur. Why is the 4' carbon important? Because it connects to the 5th. Okay. So the 5' connects here, and then the phosphates connect to the 5' carbon. All right. If we look at the bases, so here's the purine, here's the basic structure. So as you can see, there's a numbering scheme here. You don't have the prime, so if you're talking about the 7, nitrogen 7, something like that, you're really talking about, or in this case, let's say you said carbon 8, you're talking about part of one of the nitrogenous bases, not the sugar. Main thing I want to point out here, so here are the two structures, adenine and guanine. As I said, I'm not expecting you to draw these. But the main thing I want to point out is we've already mentioned that DNA methylation, we're going to talk about DNA methylation in terms of chemically modifying DNA. If it occurs in a bacterium, it's almost always going to be on this nitrogen 7 right here on the adenine. Okay. So here are the pyrimidines. Now, in eukaryotic cells, DNA methylation is nearly always on this carbon 5 on the cytosine. So it's called 5-methyl-C. So methylation is on nitrogen 7 on adenines in bacteria and on carbon 5 in the cytosines in eukaryotic cells. The other is, you already know, you have thymine in DNA, you have uracil in RNA. But biochemically, the only difference is the presence of this methyl group on the thymine. So thymine is really 5-methyl-uracil. So biochemically, they're very, very similar, except for this methyl group. Okay. And then on the phosphates, so we have the phosphate, 3 negative charges at physiological pH. So DNA and RNA is a net negatively charged molecule. So one of the things we'll have to think about. What happens when you have like charges? They repel or attract? They repel. So you guys took physics, right? At least in elementary school. So one of the things that you have happening is you got these two strands being held together, but if they're negatively charged, they're also beginning to repel each other. So later on, we'll have to talk about how the cell deals with neutralizing this negative charge on these nucleic acids. Okay, so if you just put it all together, this one, and so this is deoxyadenosine 5' monophosphate. And molecular biologists and molecular geneticists like little shorthands. And so this is DAMP. This little small lowercase d tells you that it's a deoxy. Sometimes with the ribonucleotides there will be a small lowercase r, but usually not. So the ATP that you know about as being the energy currency in cells and made in the mitochondria, is that deoxy or ribo? It's ribo. Okay. Are these nucleotides interchangeable? If I want to set up a reaction in a test tube and synthesize DNA, and somebody's used up all the deoxynucleotides, can I just throw in the ribonucleotides? No, these enzymes are absolutely specific, so you have to have them together. Sometimes you will see okay, these are monophosphates. Most of the time they're triphosphates as the precursors. And many times you will see DNTP. Which base is that? You'll see somebody sets up a DNA reaction and they have this much DNA in this buffer and 20 millimolar DNTP. What is that? What it really means is that's a mixture of all four. So when you see, it's just a very common shorthand. When you see DNTP, it just means equal molar amounts of all four nucleotides. All right. And so, and here's the thymidine this side. Okay. Any questions about that? And again, I'm not expecting you to draw the structures, but really be able to point out these key differences that I just said. Yeah.
So would there be like an R for RNA? Is it kind of
Sometimes, but it's rare.
Okay.
Most of the time, you'll just see like ATP or GTP or
You know what it means.
right. If it's not, if there's no lowercase letter in front of it, if there's no lowercase d, then you can. It's safe to assume it's ribo. Okay. So yes. Oh, you mean if I just showed you this and said, "Is this deoxythymine?" No, that's what I'm not That's what I'm leaving up to the biochemists. But I think, things like, what are the structural differences between the ribonucleotides and the deoxyribonucleotides, and how does uracil differ from thymine? Okay, the main points that I'm emphasizing now. If you want to learn how to draw those structures, that's fine. But, yes?
When cytosine deaminates to uracil, is that a favorable process for the cell?
The deamination? No, actually it's not. We'll talk about it. It's actually a hot spot for DNA damage. So we'll talk about that later on. Yeah, uracil in DNA is highly damaging. So there is a whole repair pathway that's there to find that uracil. Let's see. So actually, so what happens is, you're talking about this. So the question was, if you deaminate, or I'm trying. Okay, so if you, no, I'm just going to confuse things. We'll leave it for now. Am I going the right way? Don't think so. All right, close your eyes if you're getting dizzy. I hate it when people do this. Okay, so the main thing, we got DNA, RNA, same structure as DNA. Just to reiterate what I said, you have that hydroxyl group attached at the 2' carbon. As I said, it makes the RNA more reactive and less biochemically stable. And you have uracil instead of thymine, and again, the only difference being that methyl group. So each of these nucleotides is joined together by a covalent bond. This is the phosphodiester linkage. So the phosphodiester bond is the same in both DNA and in RNA. But it begins to show you there's a polarity here. So what's going to happen is the 5' phosphate of the nucleotide that's coming in, we're going to cover this in more detail later on, will form the phosphodiester bond with that hydroxyl group that's on the 3' carbon of the nucleotide that's already in the chain. That's why you have to have that hydroxyl group there. If that hydroxyl group is missing, then it's called a chain terminator. In DNA and RNA synthesis, will just stop at that point. All right, so let's try this question. Okay, looks like 1 minute should be enough on this one. All right. Okay, so what's the answer? There you go. All right. Okay, any questions so far? Okay. So now we want to look a little more carefully at the double-stranded structure. A very important point is that there is a polarity. These DNA strands are in an anti-parallel orientation, and there will be a number of you who are going to come into my office after the exam and be all frustrated because you didn't remember that. Yes? Which one? C? Yeah, but they're not sugars. So that's a good reason why that's not a correct answer. It's true, but it doesn't answer the question. Okay. So here's the main problem in terms of stability. We'll have to do a little quasi-thermodynamics kind of thing. This DNA molecule wants to be in its most thermodynamically stable structure. Those bases, all right, you remember what those bases, do they look like they're hydrophobic or hydrophilic? They're hydrophobic, all right? They're going to be more stable away from a water environment. So those bases are actually turned to the inside of the helix. So down the outside of the molecule, you have this alternating sugar phosphate backbone that's negatively charged. Is that going to be hydrophilic or hydrophobic? That one's going to be hydrophilic. So it actually drives the molecule into a more thermodynamically stable state by staying in that double-stranded structure. This is one of the reasons why when you denature the DNA, when you break the hydrogen bonds and separate it into two strands, you can get it to reform the complementary structure because it's actually more stable that way. So the bases are to the inside of the helix. The sugar phosphate backbone runs down the outside of the helix. As I said, you have this at physiological, so you got one phosphate at every phosphodiester bond. You got this negative charge, so you got this recurring negative charge on every phosphodiester bond. You got the two strands, you're going to get a repulsion from that charge. So to help stabilize that and neutralize that charge, we need to neutralize that negative charge. In cells, this is done by the histone proteins, at least in eukaryotic cells. So when we get into chromatin structure, we'll talk about how the histones are these basic amino acids that are positively charged at physiological pH, so they help neutralize this molecule. When we work with DNA in the laboratory, most of the time we just have to add a monovalent salt like sodium or potassium or ammonium, and that will ionically interact with the negative charges on the phosphates and neutralize the charge that way. Okay. This is a really important point. There's this polarity through the molecule. There's a 5' end and a 3' end. So if you go to one end of the molecule, if the very last atom that you see at the end of that molecule is the 5' carbon, then that's the 5' end. The 3' end will end with the hydroxyl group. So you have this polarity. As I said, it may or may not have that phosphate on that 5' end, but the 5' carbon, the last carbon of that sugar, that's the 5' end of the molecule. All right. So now we can bring those strands together, and a really important point of this is the anti-parallel orientation. So if one molecule is going 5' to 3', the complementary strand is going to go in the opposite direction. You have to keep this polarity in mind and this anti-parallel orientation, or you're going to get a lot of questions wrong on the exam. And also the molecules just don't fit together properly. Okay. So what we're trying to show here, all right, and this may or may not make sense. We talk about the directionality. This molecule is going 5' to 3' from the top of the slide to the bottom. And that's because this is the 5' end of the molecule. So there's the 5' carbon. It has one phosphate on it, but there's the 5' carbon, so that's the 5' end. And then going in this direction, it's going 5' to 3', and down here would be the hydroxyl group on the 3' end. Make sense? Okay. The opposite strand is going 5' to 3' from the bottom to the top of the slide. Okay. Also, and I'll just give you this little hint, in case you're taking biochemistry and in case they ask you to draw the bases in their paired configuration, A paired with T and G paired with C, and if you draw them as they're typically drawn on the page of your textbook, they don't fit properly. It's because of this anti-parallel orientation. So if you notice, this ribose is upside down. So you have to take one of the bases and flip it over, and then they match up. I didn't make that mistake on my undergrad biochemistry, did I? So why have I remembered it all these years? Okay, so now I just gave you a couple of points on a biochemistry exam. So we got now we're going, as I said, we're 5' to 3', but going from the bottom to the top of the page. And then here's the base pairing. AT has how many normally? Has 2 hydrogen bonds. GC base pairing has 3. This will be important later on as we talk about the stability of the molecule, but in certain situations, the cell has to break those hydrogen bonds and be able to open up that helix. It's got to be able to do that under physiological conditions. So it's not surprising in many of those places you find a lot of AT base pairing and little GC base pairing because it takes less energy to break the 2 hydrogen bonds than to break the 3. Okay. So we got that, GC base pairing. There's the phosphodiesterase linkage. Okay. And over here is the RNA. Is this one going from top to bottom? It's going which way? If you can read, 5 to 3. Same thing. Even though it's RNA, works the same way. Here's the 5' carbon, so there's a 5' end. Down here, do you have a 3' hydroxyl on RNA? Yes. You also have it at the 2' carbon. So if I showed you this molecule and one of these strands, you could tell me which one was RNA and which one was DNA? Good. All right. Oh, a few more boxes in there. Okay. Which really brings us to a very important point, not only for DNA structure, but for cellular processes in general, and the importance of weak forces. So you have a lot of, whether you're looking at double-stranded DNA, that the hydrogen bonds have to be broken under physiological conditions. RNA, we'll talk about later on, actually has a tertiary structure and forms hairpin loops and stem structures and such. Or even proteins which have to have their proper three-dimensional structure. You have these weak forces, electrostatic interactions, van der Waals interactions, hydrophobic interactions, hydrogen bonds and such, which work over short distances to allow a high level of specificity, but also are weak enough, they're strong enough to hold a molecule together under physiological conditions, but weak enough to break them apart also under physiological conditions. So one of the most important for DNA is the hydrogen bonding. So you've probably seen this from chemistry, just a little bit of a review in terms of what's happening in DNA. I don't know, I kind of think of them like little molecular magnets. So you have an electronegative atom such as oxygen is a good one. A lot of times it can also be nitrogen. You got electrons being shared between this hydrogen, this electronegative atom. The electrons are going to spend most of their time around the electronegative one, and you get this dipole moment where you have a very small positive charge here and a small negative charge here. That allows it to interact with the negative charge on the next molecule. So in these water molecules, should water be a liquid at room temperature? If you compare it to other molecules like CO2 and such. No, it's a liquid because of the hydrogen bonding. It forms this lattice structure and has enough stability to actually be a liquid at room temperature. So these hydrogen bonds, so keeping in mind just an approximation here, they're not very strong. A single hydrogen bond is only about 5% the strength of a covalent bond. But because you got thousands, if not millions of these bonds coming together, there's this cooperative effect, which gives it a very stabilizing force, yet can still be broken apart under physiological conditions. Okay, so we already covered that. All right. So the GC base pairing, as we said before, is more stable than the AT. All right. and we have this complementary structure. And this was actually an important point that came out of Watson and Crick's paper, is once they recognized this complementary structure, it also suggested a way that the molecule could replicate itself. As long as you had one of the two strands, then you could duplicate that double-stranded molecule. So in that one-page paper in Nature, not only did they describe the structure of DNA, but even suggested a mechanism for DNA replication. Now, by convention, here I've designated the 5' end and the 3' end. By convention, if that's not marked, if that was just AAC, TTG, CGC, then you can assume that that's the 5' end on the left, and it goes 5' to 3' as we draw the molecule. Excuse me. Now, I'm going to put this on the exam, and I'm going to give you this sequence, and then I'm going to ask you what the complementary sequence is, and I'm going to have 5' to 3', and you're going to tell me it's TTG, AACG. Okay, and why is that wrong? You have to watch the polarity. Okay, so draw it out if you have to, but keep in mind the anti-parallel orientation. And this will apply to everything that we're going to be talking, even if tRNA molecules interacting with the messenger RNA, it's always an anti-parallel orientation. So when you put all this together and kind of show in cartoon form what Watson and Crick put together, is you get what's typically called bDNA. So I'll show you an example. There's A, B, and Z. So this is B-form DNA. We think this is pretty close to what the DNA looks like inside cells. So you have a perpendicular axis running through the center of this molecule. The bases are inside the molecule, and they're oriented with each other, and they're almost, I'll show you why not, they're almost but not perfectly perpendicular to the center of the axis. There is about 10 nucleotides per turn of the helix. So if we start at one point, as we go around the helix, come back around, it's more like about 10.4, but we'll call it 10 for our purposes. The diameter is fairly uniform with a diameter of about 2 nanometers. All right. So those are the general dimensions. Now, if you look at this space-filling model, what you see is there's actually a minor and a major groove that forms in the double helix. The reason these two grooves are not exactly the same size is because those bases are tilted slightly. The grooves would be the same size if it was perfectly perpendicular to the center axis, but they're tilted slightly. So you have the main groove and the minor groove. Now, you look at this space-filling model of the DNA, and one of the recurring themes that we'll keep coming back to as we go through the remainder of the course is protein interactions with a specific DNA sequence. Transcriptional activators, lots of different examples. The bases are inside the helix, all right? How could the protein read the DNA sequence without unwinding the helix? Or is it going to have to unwind the helix every time it binds? Yes. Yeah, pretty close. The key point here is the base of this major groove is actually the top edge of the bases. So the proteins can fit into the groove on the DNA and literally read the DNA sequence. So when we start talking about these DNA binding proteins, you see certain recurring motifs or substructures in that protein. One of the first ones was called zinc fingers because they look like fingers that are coordinated with a zinc atom, but they're exactly the right molecular dimensions to reach down into the groove and actually read the bases. So the DNA binding proteins can read the protein sequences without having to unwind the helix. The other two forms that I'm not going to spend a lot of time on, this A form is sort of a shorter, fatter version. As far as I've ever been able to tell, this is really what more like dehydrated. So this is a fully hydrated form. If you dehydrate it and remove a lot of the water, you get this A-form structure. It's in the literature. I'm not sure it really has a biological relevance. The one that is interesting is Z-form DNA. So DNA, the B-form DNA that as we think it occurs in cells, is a right-handed helix. So if you want to envision that, if you take your right hand, you point your thumb down, the way your hand naturally curves is the direction of a right-handed helix. The left-handed helix goes in the opposite direction. So the Z-form DNA was rather interesting because it literally flips into a left-handed configuration, and it got its name because if you trace the backbone, the sugar phosphate backbone through it, it follows a Z configuration. Right? For a long time, it was thought that it probably had some biological role in promoters and such. That seems to have been disappearing from the literature, so I may not be up on the most recent papers on zDNAs, but I would put it this way, it's questionable as to whether zDNA actually has a biological role, whether it really exists in DNA. It was created first artificially in Alexander Rich's lab when they were playing around with the DNA, and they put it in a very high salt concentration, and then looked at it with the X-ray crystallography, and it had flipped into this Z form, and there was obviously a publication there. There's a certain DNA sequence, I won't go into the details of it, that biologically should form zDNA, but I'm not really sure there's compelling evidence that it's biologically important. The main one we're going to focus on is the B-form DNA, which we think is most relevant biologically. Any questions? Okay. Just to point out some of the subtleties, DNAs are the way we've drawn it here, it's a very regular looking molecule. So even though these proteins can find these DNA sequences, are there some other landmarks that it can use? Well, as people have looked at it a little more carefully, there are some very small but clear variations in local differences. What am I trying to say? In different locations within the DNA molecule, depending on the sequence, on other molecules that might be associated with it and such. There are some small minor variations. And some of these small topological changes may in fact be sufficient to help these proteins find these DNA sequences. All right, so let's try this question. I'll go to 1 minute and 15 seconds on this one. Okay. 3, 2, 1. All right, what's the answer? E. Okay. Is D ever correct? No? Never? Maybe. When would it be correct? In fact, this was part of the problem Levine ran into. He happened to be using a DNA sequence that was close to being 50% G plus T. If he had chosen a few other sources, then he might have gotten different results, but it also may have been just the uncertainties in his measurements because of the instruments that were available at the time. Okay. So at the, yes. Well, you could have a 50% G plus C content. Some organisms have that. But it tends to range from about 30% to 70% depending on the source. And you can't predict. You can't say, well, bacteria have low G plus C contents and humans have high. It just...
What was he studying that was high?
Oh, he was probably just getting it from some typical, the insect cells or whatever that were widely available at the time. I think E. coli is around 30%, but it also is, the technology it was using may not have been sufficiently. At that time, trying to do quantitative biochemistry would be like trying to repair a NASCAR with a hammer and a pair of pliers. I mean, that's about the best tools you had available. All right. Yes. Yes. The question is, yes, the phospholipid bonds, as far as I can know, and I've never read any exceptions, doesn't matter whether it's a virus, RNA, DNA, prokaryotes, eukaryotes, biochemically they're identical. So with all the differences there are between the genetics of a human cell and of E. coli, at the very basics, the double-stranded DNA, the genetic code, all of this is very universal. Okay, so at the beginning of the chapter, we laid out these three requirements that we were going to have for the genetic material, and why protein really doesn't fit the requirement. But if you look at Watson and Crick's model, you can see that it does in fact fit all three of these requirements. One was Levine was way off base with his model because he just had this simple little circular model, this model for this DNA. And the question was, how could you encode the genetic material? How could you encode the information that had to be stored there? Well, with Watson and Crick's model, it became clear it was the sequence of the nucleotides that provided the primary information. I'm waffling on that a little bit and saying that it's the primary information because, as we'll see as we go further into the course, there's a lot of other subtleties to the DNA that are providing genetic information that go beyond just simply the nucleotide sequence. But at the basic level, it was clear that you could encode this genetic information in the sequence of the nucleotide. So that's the primary structure of the molecule. So that meets the first requirement. The biggest problem with the protein model was, how do you have a mechanism to precisely replicate the molecule? So we have these 20 amino acids for proteins, and people are trying to come up with all kinds of models and templates and such. But in fact, the complementary nature, the fact that you got the A paired with T, G paired with C, immediately suggested that one strand could serve as a template to replicate the other. And that's what they suggested. Now, what they didn't know at the time was exactly how that molecule worked. And that'll come up a little bit later in another one of these landmark experiments. But you can now replicate the molecule, so that fulfilled the second requirement. And at third, the genetic material must encode the information to give you the phenotype. So the phenotype is going to have to be based on some gene product. That's usually the protein. So part of the problem was, especially in eukaryotic cells, the genetic materials in the nucleus, the ribosomes, which are responsible for synthesizing the proteins, are out in the cytoplasm. How do the two communicate with each other? And in fact, it was Francis Crick who first suggested messenger RNA as this transient molecule that would then serve as the template to bring the information from the DNA in the nucleus out into the cytoplasm. So that's where we have this RNA intermediate. So I'll use this slide because of what I thought was an easy question on the exam, but ended up being a confusing one. So let's just be clear. The original central dogma that Francis Crick put forth is what's shown on the left. And that is, there's this unidirectional flow of genetic information. It's stored in the DNA. There's a temporary transient copy of it in the form of RNA. That's through the process of transcription. And then that RNA serves to direct the assembly of the amino acids to give you the proteins that are made by translation. Everything was fine until the discovery of the retroviruses, which do in fact allow reverse transcription. In fact, you even have replication of the RNA viruses. But if we take the viruses out of the equation, then the central dogma remains unchanged. Okay. Any questions so far? All right. Okay, so special structures. The double-stranded DNA, that's where it gets a little confusing. So the double-stranded DNA, pretty straightforward. It's got a lot of constraints on it because of the complementarity and the double helix. So there's not a lot thought to be important in the three-dimensional structure. But single-stranded DNA, and that's what this little SS indicates. So sometimes you'll see DS for double-stranded or SS for single-stranded DNA. And especially RNA is normally in a cell, is RNA a double-stranded or single-stranded molecule? Well, it's normally single-stranded. But what we're learning is it takes on a rather elaborate three-dimensional structure to it that's all sequence dependent. And that's what we want to look at here. So what kind of structures can you get when you're looking at a single-stranded nucleic acid? Is there a question? Okay. This is a really important sequence, structure, whatever you want to call it. It's confusing, and I'll walk you through it, and hopefully you'll be able to see it. It may not make sense at first. It's an inverted repeat. What it means is the sequence is exactly the same on both strands when read in the same polarity. And these inverted repeats pop up all the time in terms of being regulatory structures and controlling elements and parts of transposable elements. So you see this in molecular genetics time and time again. So we have a sequence. We got 4 bases. These 3 dots just indicate some other sequences between it. And then we got these 4 bases. If we read this 5' to 3', we have 3 A's a G. If we go to the opposite strand, now we have to come back right to left. We got 3 A's and a G. Okay. And now we have C and 3 T's, a C and 3 T's. So inverted repeats are the same. You can recognize them because they're the same sequence on each strand, but you have to read them with the same polarity. The reason they're important in single-stranded molecules, especially RNA, is because they give a 3-dimensional structure to the molecule. So they can form what are called hairpin structures. So here's the same thing we just saw. The reason they're important is they're self-complementary. So you also see that this A is complementary to the T. So if you start at the A and the T and you work out, what you see right here is they're self-complementary. So you can form these double-stranded structures within the single-stranded molecule. For RNA molecules, it's not the sequence that's important. It's the secondary and tertiary structure it takes on because of its sequence. All right? If you have a space between, if you have nucleotides that aren't part of the inverted repeat, then you get this loop structure. Okay. Now, why is it sort of pushed out into its maximum configuration? Where are the bases? Are they protected from the water? No, they're actually forced out into the water. So the molecule is taking on its most stable structure. So these inverted repeats give you these stem loop structures. If you don't have a few nucleotides between them, then you can get these stem structures. It's the same thing, except now the inverted repeat is perfectly complementary, and there's no nucleotides between it. But as this becomes more complex, and you can take on all sorts of different hairpin structures and loop structures and such, this is where the RNA takes on its tertiary, or basically its three-dimensional structure, and this is what gives functionality to the RNA. This is becoming more and more important as we're discovering more, especially small microRNAs that are carrying out functions, genetic functions in the cell that were completely unknown just a couple of years ago. For instance, the junk DNA, the dark matter of the human genome, which makes up about 98% of the human genome. Francis Crick, again, coined the term junk DNA, and for many decades it had no known function, but now with genomic sequencing, there was a large project called the EDGE project, published, the results published about 3 years ago, and it looks like this junk DNA actually codes for about 4 million small little microRNAs that we didn't know about. And so now maybe 98% of our genome is important to regulate the expression of 2% of the genome. Yes?
What's going on in the structure right there? So there's like a loop that intersects two lines to the left.
Up and left.
Right there.
Right. Oh, here?
See how that's,
Oh, this?
yeah, up and down.
I think some artist got carried away with the pen.
Are palindromes the same way?
Very similar. What do you call them? A lot of times you think about palindromes in terms of like a restriction enzyme sequence and such. Yeah, technically they would be. They would form inverted The difference is, and nobody's clear if this really works, and when you have palindromes, potentially you could actually split into two opposing hairpin stem structures. You could.
Oh, you can. Okay.
But especially if that's in double-stranded DNA, thermodynamically it's not likely to happen, but theoretically it could. So the palindrome, you really need the two strands. The inverted repeat, you're recognizing it as two strands, but you're just using one strand to form the inverted repeats that'll give you the hairpin structure. Yes?
So this is, we're going with RNA primary, secondary structure, right?
Yes.
It's different from the concept of DNA primary, secondary structure.
Yes. Okay. For DNA, both of them, the primary structure is the sequence. The secondary structure is different because DNA has this complementary double-stranded structure that is most stable when all the hydrogen bonding occurs, so that three-dimensionally it's a fairly uniform molecule. Here, the molecule is single-stranded, so the structure it takes on really depends on the sequence of it. Another one just to start to introduce this idea of DNA methylation. We're seeing this more and more examples of these chemical modifications. Now, these chemical modifications occur after DNA synthesis. There are not nucleotide triphosphates floating around in the cell that have methyl groups on them. So the DNA is synthesized first without the methylation. The methylation comes in later. So again, this is one. So here's 5-methyl-cytosine. This can kind of go to you asked the question about the deamination. I think this figure shows it. This ends up, I'm jumping ahead, but this ends up being a hot spot for DNA damage because if you deaminate it, if you remove this amine group, you end up with 5-methyl-uracil. So the difference between cytosine and uracil is this amine group. Well, 5-methyl-uracil is thymine. So the DNA repair systems can't see it as a damaged DNA molecule because it inadvertently is converting a C into a T, but it's biologically recognized, so they tend to be hotspots for DNA damage. Yes?
Is an idea of epigenetics the fact that we can do things in life that methylate DNA or like demethylate it? Is that like
That's part of
the
it.
idea? Part of the
That's
idea?
part of the story. I mean, epigenetics is a brand new story,
Yeah.
but it looks like, and the idea of DNA methylation has been known for decades, but I think the general idea was that as you go through meiosis, all that methylation was stripped off of the DNA, but now it looks like maybe not. That some of that DNA methylation is, and that's the whole point of the epigenetics, is there's some chemical modification due to something a parent did, and that's getting transmitted through the gametes to the next generation. That's how we think it works.
Okay,
It's
so
a it's a whole no, it's a whole new area. Did you have you have a question? Okay. All right. And now to make it even more interesting. All right. And what we're going to in the next chapter is to getting into higher levels of organization of this DNA and the chromatin. Now, I said DNA binding proteins can read the DNA, the double-stranded sequence. They can bind into the grooves, especially the major grooves. They can literally read the base sequences. But some processes within the cell, DNA replication and transcription, require that the helix has to be, the double-stranded structure has to be broken. The hydrogen bonds have to be broken in order to give access of the polymerases to those bases. And that's a thermodynamic problem. How do you put enough energy into the system to break those bonds under physiological conditions? Well, one of the things that we found now is these proteins, for instance, this, I think this is probably DNA polymerase, these proteins can bind to the DNA and literally bend it as they're binding. That puts a torsional stress on the DNA and helps break those bonds open. So we can also get shaping events to the DNA as a result of proteins binding to it. Especially in eukaryotic cells, this protein-DNA interaction is really what has to be thought about. Okay. So that was amazingly few questions, which means I'm either really good at explaining this or you're really confused at this point. Okay. So while you think about whether you're confused or not, let's try this question. Details, details. It's always in the details. All right, there we go. I'll go to 1:15 on this one. I don't know what it is. I just make this up as I go. All right, so what's our vote? All right, so what do we got? We got a new virus that infects your hot tub, hot springs. The viral genome is 4 ribonucleotides. So right there can be DNA. We already told you that. So it's 8% A, 42% C, 42% G, and 8% U. The genome is most likely double-stranded because why? Because you got equal amount, but it doesn't have to be. Okay, good. All right, so this slide, this formally ends the material for the exam on next Wednesday. I'm going to start chapter 11. Let's see, I've got 92 slides left. Actually, it runs into chapter 12. Don't worry about it. So it's an extension of what we're doing here. We're now going to look at higher levels of organization of chromatin, specifically looking, at least most of this is going to be in eukaryotic cells. How does all this massive amount of DNA get packaged into cells? We've got to talk about things like supercoiling and torsional stress and such. So you won't have to figure this out until exam 3. Okay? All right, so this is chapter 11. Oh, that's right, organelle. Okay, and this also has the chapter on organ. I forgot to reorganize, so we're not going to get that far today. Organelle DNA, so we'll be talking about the structures of mitochondria, chloroplast DNA. But that's later on. So here's the problem. As I pointed out at the beginning of the course, was these DNA molecules are huge. And they have to be, this massive amount of nucleic acid has to be packaged into the cells in a fairly small amount. So even in the bacteria, which have fairly small genomes, and I've shown you pictures where the DNA is just spewing out, there has to be some way of compacting it. In bacteria, it's not that detailed. Basically, you can imagine the genomic DNA, it's a circular genome, forms what we call a nucleoid. In most cells, bacterial cells, it tends to be associated with an inner cell membrane. It's got a little bit of protein associated with it, but not nearly as much as what we see in eukaryotic cells. So again, the main emphasis on this chapter is how do we package this DNA in chromatin, which we find in eukaryotic cells. Okay. So I got enough time here. Let me see if I can go through this and see if I can make sense out of this. So we've got we have the helical structure of the double-stranded DNA. We just talked about that. It had how many base pairs per turn? It had 4, 10.4, 10, one of those numbers. Okay, we'll use 10. So normal, relaxed DNA is going to have 10 base pairs. So if you start at one point on the helix, you go around the helix, come back to the same point directly above it, it's 10 base pairs. Okay. What we got to talk about here is supercoiling. In cells, you don't have it in what we call this relaxed form with the 10 base pairs, but it's under a torsional stress. I'll try to explain that in a few minutes. And to compensate for that torsional stress, it has to supercoil. So in its most relaxed state, that's where we'll start. This B-form DNA, which we think is the most biologically relevant form of DNA, will have 10 base pairs per turn. So we have a small little molecule. It's 100 base pairs of DNA, and it's a circular molecule. It's going to have 10 complete turns of the helix, right? Okay. Now, back in the dark ages, you guys don't know this, phones used to have cords on them. You know, or maybe you've gone to grandma's house. I don't know, my grandmother passed away years ago, but she had the same black AT&T phone. It could have gone through a thermonuclear war. I mean, they just were indestructible. And they had these little coiled cords on them. And so we've used them for years as a model for the superhelical structure of DNA. So that's why the author is trying to do that here. Over here is an electron micrograph of a relaxed circular plasmid molecule. So you can actually visualize DNA molecules very nicely with the electron micrograph. And here it is in a cartoon form. So here's the relaxed circular DNA. If it's a circular DNA molecule and it's fully relaxed circle, it's a flat, planar molecule, and it would lie flat on that table. Okay? Kind of like an inner tube or whatever. Or think about it like as a rubber band. But that's not the way it exists in cells. Cells, there is a torsional stress that's put on them. Much of it is to help compact the DNA. So if you take a rubber band or one of those hair things, whatever, okay, the relaxed circular part, and you hold on to it and you just start wrapping it up. You're supercoiling it. And what happens to it? It gets all knotted up and it gets much smaller. So it's a way of helping to compact the DNA into a smaller space. But we're going to put energy into winding up that DNA. Where here we're actually going to be changing the number of turns. So it's like taking a spring and turning it, and we can either add turns or remove turns from it. If we do that, the molecule has to do something to compensate for that stress. So as we put this torsional stress and begin to rotate this molecule, it's going to flip into a large supercoil. That flat planar molecule is going to flip on itself. If we over-rotate it, by over-rotating means we're going to add more, we're going to have more than 10 turns, more than 10 bases per turn of the helix. We will get positive supercoils. I'll show you a figure of that in a minute. If we under-rotate it, so we have less than 10 turns, then we're going to get negative supercoils. This is what we find in cells. We don't find positive supercoils, we find negative supercoils. Okay. So here's our model that we started with. And here we're going to add 2 turns. So if you look closely at this, you can see the spacing between these turns is a little closer. That's what they're trying to show you. So now you've added that the DNA is actually wound up tighter than you would expect at 10 bases per turn of the helix. So this is now over-rotated, and it's going to cause positive supercoils. This one is under-rotated, so now you've turned the helix back in the opposite direction. You have fewer than 10 base pairs per turn. But what do you notice about it? Part of the double-stranded structures come apart. So one of the reasons that cells are going to favor negative supercoils is they can actually take advantage of this torsional stress to make it easier to separate the DNA strands when they have to. For instance, this has to be the first step that happens before RNA synthesis can occur. And this transition from the closed to this open complex is actually the rate-limiting step for transcription. So this is why it's believed you can get the same result. Positive supercoils, negative supercoils. The outcome is the same, but there are certain biological advantages to going with the negative supercoils. And this is what you find in cells. So that's what it's trying to show you here. These 2 structures look pretty much the same. The difference is the direction of it. So as I said, DNA, just B-form DNA, is a right-handed helix. So that's your thumb pointed down of your right hand, and you're going in this direction. The positive supercoils will flip or supercoil in the same direction. So they're also going right-handed. The negative supercoils, when it's under-rotated, flip in the opposite direction. So if you look, this strand is crossing over the top here and behind this one, and it's the opposite over here. So you still get supercoils, it just depends on which direction it flips. Okay. Does that make sense? Can you visualize that? Yes.
So can you repeat that? The supercoil or something?
Right. So what if we started here? So here's the relaxed circle. This is the 10 base pairs per turn. Now, I kind of think of this like let's say it was a coiled spring. If you wind it up, if you have actually more than 10 base pairs per turn, you've added turns to it, and it's over-rotated. And that's going to give you supercoil. So either way, anything that deviates from that relaxed circle is going to cause supercoiling. If you over-rotate it, it's going to give you positive supercoils, which means the positive just means the supercoils go in the same right-handed direction that the helix is turned. Negative supercoils just flip them in the other direction. You're turning one way, it goes this way. You turn it the other way, it flips back in the other direction. Okay? So this is what we find in cells, is this negative supercoiling. Yes?
Wouldn't it be
be more space conserving using positive supercoils though? Since it's such a big molecule inside the cell and they have little space.
Well, you get this. The outcome's the same. You're still getting the coiling, but the negative supercoiling is you're taking also advantage of the fact that you're actually destabilizing the helix a little bit. So especially if this sequence is AT rich, then it's going to be the first one to kind of pop apart from doing this. This is probably not exactly what's happening in the cells, but it's showing that you get the same outcome in terms of compacting the DNA. But by using the negative supercoils, if you do positive supercoils, it's going to make it even, you got to put even more energy in to breaking the hydrogen bonds and opening up the helix. So it'd be even more work. Okay. So if that makes sense, let's try this. I think I got a couple of questions here. So. I'll go to a minute and 15 seconds. Let's see. So the 3 students who answered D must have just walked in. All right. What's really the answer? Okay. This one might be a little more challenging. Let's try this, see if. We can go to a minute and a half on this one. All right. Let's see how. Oh. So what's the vote? What? Okay. You got 300 base pairs. If it's a relaxed circular molecule, how many rotations would it have? It would have 30, 10 per turn. But it's only got 20, so it's under-rotated, and this molecule will have negative supercoils. Okay? Now, here's an important idea. What I didn't say, but I only implied when I was telling you this, is we're not, when we're doing this supercoiling and rotating and under-rotating and over-rotating, we're not breaking any of the covalent bonds. We're really just sort of slipping the hydrogen bonding. The question is, what happens if the circle gets broken? All right. Well, there's an idea called linking number, and I'm not going to bore you with that. Okay? But here's the important idea. How many of you have thought about what happens during DNA replication? I didn't think so. Okay. We talk about DNA replication. You should at least know that the two strands have to get unwound. They never come back together again. One strand shows up in each of the two new daughter helices. So as that DNA is unwinding, what's happening is all these supercoiled knots are forming ahead of the complex, because that torsional stress has to go someplace. So it's unwinding the DNA. The DNA ahead of that replication complex is knotting up. If there isn't some way to fix that, if it gets too bad, it's going to jam the whole system up and DNA replication will end. So we need a group of enzymes that can go in there and change that superhelicity, that can either remove or add negative supercoils or positive supercoils as they're needed. Okay? Now, what can happen is, another important point, we have this circular molecule, and let's say it's the one that we just talked about, the one in the question, and it's got these negative supercoils. That also assumes that every single covalent bond on both strands is in place. But we can nick the molecule. The nick is just a jargon term meaning one of the phosphodiester bonds on one strand is broken. When that happens, because the molecules are just being held by these hydrogen bonds, the strands can literally unwrap themselves and go back to the most stable, relaxed configuration. So if we break a strand, we can actually change the level of supercoiling. Now that can happen by accident, or we can do this with enzymes, which I'll tell you about in a moment. Another important point is linear double-stranded DNA cannot be supercoiled. Does that make sense? You just have a linear molecule, you rotate one end, the other end turns. But we also said that the DNA in the chromosomes of eukaryotic cells is linear, and it is supercoiled. Well, you can supercoil linear DNA if you constrain one end. So if I hold on to one end of the molecule, I can turn the other one. So I'll show you, may or may not get to it today, but a figure where even though the molecule is linear, it's constrained into loops, which effectively make it a circular molecule. So these enzymes that can add or remove these supercoils by removing rotations from the DNA helix collectively are called topoisomerases. So they're changing the topography of the DNA molecule. In general, there's a whole range of them. We're not going to go into the details of it. In general, what they do is the enzyme binds to the DNA. It cleaves one of those phosphodiester bonds, just like making the nick that I talked about. And literally, while the enzyme is still bound to the DNA, it passes one strand around through the enzyme, back around, and reforms the phosphodiester bond. By doing that, it's changing the number of rotations on the molecule and thereby changing the superhelicity of the molecule. Just some general categories of them, and mainly I want you to understand that topoisomerases are there to change the level of superhelicity either by putting in negative supercoils or positive supercoils what's needed. There are a couple of the enzymes, kind of Topo 1A is a prokaryotic enzyme that only relaxes negative supercoils. So would it add or negative supercoils or positive supercoils? If it added negative supercoils, it'd wind it up even tighter.
No, it's possible.
So it has to put positive supercoils in to relax the negative supercoils. Topo 1B is a eukaryotic enzyme that can either relax the DNA or even add positive supercoils to it. Topo 2, the main difference between the type 1 and the type 2 is whether they only break one strand of the DNA or break two strands of the DNA. But the end result is pretty much the same. So the take-home message is you have this category of enzymes, the topoisomerases, which are required for controlling the level of supercoiling in the DNA molecules. Would a mutation in topoisomerase in E. coli probably be lethal? It is. Why is it? When it has to go through that DNA replication, if you can't, you're basically creating positive supercoils ahead of the duplex. If you can't remove those, the whole system jams up and DNA replication stops. Okay? All right. This is probably a good point to stop then. Okay? So today must be Wednesday. All right. So, as I said, we've now finished all the material for exam 2. That ended with chapter 10. This material from chapter 11 will be on exam 3. Otherwise, enjoy the remainder of the week, and we'll see you on Monday. Come in with questions on Monday.